Hey, Mr. P here. In this video, we're going to talk about how plants transport phloem sap through the plant. So we're talking kind of the other side of the vascular tissue, that is the phloem. In the last video, we talked about the xylem specifically and what the plants use xylem, how they structure xylem. If you haven't seen that one, go check it out. Otherwise, in this video, like I said, we're going to talk about what the phloem specifically is used for and how plants structure the phloem in order to function accordingly. Let's go. So, plants transport organic compounds from sources to sinks, and when I say source, that ultimately sounds like the beginning or the origin, and sink ultimately the final destination. So this is how plants transport organic compounds being glucose and some of those other organic products of photosynthesis, from where they're produced to ultimately where they're stored. Okay? Sources need to be thought about where things are produced, and sinks are where they are stored. So, from where they are stored, or from where they are produced to ultimately where they are stored. Phloem is mostly made up of these sieve tubes. Okay, so just like xylem, it is a continuous tube from ultimately the leaf tissue to the roots. And it is made up of these cells that are alive, which is different than the cells that are going to make up the xylem. Remember that xylem is composed of kind of remnants, the cell wall remnants of past live cells. They are no longer living. And so these cells that are going to make up the phloem are going to be made into these sieve tubes, and they're going to be associated with companion cells. And these companion cells are exactly what they sound like. Companion cells are going to be the companion to these phloem cells, or these phloem sieve tube elements, and they are going to kind of be life support for the cells that ultimately make up the tube itself. They are going to be connected via a sieve plate, which you can see right here. This is the sieve plate, otherwise known as the sieve tube plate, and they're going to be extremely rigid. These sieve tube plates are going to kind of structure and provide structure to these sieve tube, or from these phloem tubes from the inside, so that they can withstand very high pressures, which are going to be needed in order to kind of generate this hydrostatic pressure gradient from the source to the sink. And a little bit more on that to come. But if you remember, the xylem are going to be very low pressure. These phloem are actually going to be high pressure, and that's because we have phloem sap that is constantly being produced in the leaves and is being pumped kind of into these sieve tubes in order to increase the pressure so that the phloem sap can actually flow throughout the plant. Sieve plates have these pores in them. They allow water to move, and they allow the dissolved organic compounds to move throughout the plant. Remember that phloem is going to be much bigger in terms of the internal diameter, or the lumen size for the actual physical vessel, than the xylem. Xylem are going to be much smaller because xylem, as you recall, are transporting just water molecules. Water is actually going to be the majority of what the phloem transports as well, but water in the phloem sense is the solvent that the solutes are dissolved in. So water is still going to be found in the phloem, but it's not going to be just water. It's going to be phloem sap, which is a lot stickier, a lot more viscous than true water, but water is going to ultimately be the solvent that the solute is dissolved in. These companion cells, which are the cells that are found on the kind of exterior or the periphery of these companion or the sieve tube elements, are going to be connected to their sieve tube members by plasmodesmata. You can kind of see them here. They're basically pores that allow water and other organic compounds to move through. Again, these cells are the life support that are going to provide all of the things and the energy that these sieve tube elements are going to need in order to stay alive, because phloem cells are living, whereas xylem cells are non-living. Okay, the movement of materials is always from source to sink. Remember, source is where they are produced, to sink is where they are stored. This is called active translocation. It is organic molecules like sugars that are produced via photosynthesis. They are dissolved in water, and the solution is called, therefore, phloem sap. If you were to extract phloem sap from a plant, it would be very viscous, it would be very sticky because it has a very high sugar content. Okay, that is the food source that plants provide themselves via photosynthesis and ultimately store until they are needed to produce an active amount of energy, which is ATP. The source is a plant organ that is a net producer of sugar, that is photosynthesis or hydrolysis of starch. That would be a leaf. Okay, so source cells are going to be where these photosynthetic products are made, which is the leaf. And a sink is the plant organ that uses or stores sugars, which is typically always found in roots, buds, stems, seeds, fruits. Things that are not actively photosynthesizing, but are actively maturing, actively growing, actively developing, and require a large amount of glucose in order to produce a large number of ATPs. So the pressure flow hypothesis, this is the way that plants are believed to move or translocate these sugars from source to sink. And so there is a loading phase. The loading of the sugar into the sieve tube from the source, again from the leaf, is reducing the relative water concentration in the sieve tube members, which is causing osmosis from the surrounding cells. So the loading phase takes place at the source. The plant photosynthetic organ, which is the leaf, is going to produce a large number of these kind of sucrose or glucose molecules. They're going to be loaded into the phloem, which is going to decrease the water concentration because it is increasing the solute concentration. It therefore causes osmosis to move in terms of water from its high to low, which causes the pressure to increase. Okay, the uptake of water at the source causes a positive pressure, referred to as hydrostatic pressure, in the sieve tube, which results in the flow of phloem sap. So we load the sugar. It causes a lower water concentration or a higher solute concentration. Water is going to move in via osmosis, creating an increase in pressure kind of upstream from the phloem, and therefore net movement down the plant from high pressure to low pressure, where the sugars will ultimately be forced into the sink, which is likely the roots or fruits or seeds or berries or something like that, and that is ultimately where they will be stored. Okay? This hydrostatic pressure is diminished by the removal of sugar from the sieve tube at the sink. The sugars are changed into starch at the sink, and since starch is insoluble in water, it exerts no osmotic effect because they are therefore not solutes at that point, because they are not dissolvable in water. Because we are pulling the solute out and causing it to turn into starch, it is increasing the water concentration here by decreasing the solute concentration, and therefore osmosis will occur in the opposite direction back into the xylem, which increases pressure here and follows the normal transpiration flow of water up the plant as the water is both pulled out of the xylem due to that hydrostatic pressure, but also is released as water vapor through transpiration out of the plant or out of the leaf. Okay, xylem recycles the relative pure water by carrying it from sink back to source, and it flows back into the phloem, or can be lost, like I said, as water vapor through that transpiration stream. So phloem transports water and solutes using that hydrostatic pressure gradient. We just kind of talked about that, but there are a variety of other transport mechanisms at play within the plant, and so these are a few of them. So there's active transport, which is loading and unloading of sugars from source to sink. It requires proton pumps and specialized membrane proteins called cotransporters, which can be seen throughout the phloem. Basically, as the leaf produces the phloem products, right, or as it produces the photosynthetic products, sugar, it's going to actively pump those sugars into the sieve tubes in order to move them from source to sink. There is a passive transport, so the movement of phloem through the sieve tubes, that is a hydrostatic pressure gradient that is moving from source to sink passively, does not require active transport, doesn't require ATP. This hydrostatic pressure gradient is produced by the compression of a liquid in a confined space or by the addition of solute particles to a liquid in a confined area. Since water is relatively incompressible, adding solutes to a limited space filled with water increases the pressure. So like I talked about in that last slide, if we have sugar that is constantly being loaded into these sieve tubes or these phloem tubes, it's going to increase the pressure at the top of the plant, and because the phloem is taking out the sugar, or the sugar is being moved out of the phloem into the roots where it is stored at the sink, it's going to have decreased pressure, and so the phloem sap is going to move from high pressure to low pressure from leaves to the sinks passively, without energy. Water and solutes will always move from an area of higher pressure to an area of low pressure. That is a given. Water moving obviously would be osmosis. Solutes moving would be regular diffusion. Okay? Now, how do we know how plants utilize sugars, and how do we know how these sugars move throughout the phloem? Well, there are experiments, and there was a very kind of important experiment, important discovery that utilized these aphids and aphid stylets in order to kind of track radioactively labeled carbon dioxide. And so the way that the experiment worked is that researchers took a plant and they took these aphids, which are little insects that are naturally penetrating phloem with their stylet, which is a kind of a piercing mouth part. P is labeling the phloem, so this is the tubes that would be used to move phloem sap through the plant. And researchers took a radioactive carbon dioxide and they localized or isolated on one particular leaf that radioactive carbon dioxide. So all of the sugars that this particular leaf used or produced via photosynthesis would have radioactively labeled carbon. All the other parts of the plant, all the other leaves, were exposed to the normal form of carbon dioxide, which is just abundant in the atmosphere. And so they were able to therefore track the movement of solutes from this particular leaf throughout the plant. Did the phloem transport solutes from this leaf and move them everywhere in the plant, or did they stay localized from source to sink? That's what they wanted to find out. Okay, what they did was they let this plant radioactively produce they allowed the plant to photosynthesize. They gave this particular leaf only the radioactive isotope carbon-14, or carbon dioxide that utilized the radioactive isotope of carbon-14, and they put a aphid here, and after they put the aphid here and allowed the aphid to insert its stylet into the phloem, they took a sharp scalpel and they severed the stylet, meaning they cut it, removed the aphid, and therefore were able to analyze the phloem sap that kind of came out of the plant at that one particular area. So when they severed the stylet of this aphid, they then were able to isolate the phloem sap of this particular area, and they determined that it had sugars that had radioactively labeled carbon. They then put a stylet, or inserted an aphid here, severed the stylet, and they found that when they identified the phloem sap, it too had it there. They continued this all the way down the plant, allowing the aphids to insert their stylet into the phloem. They severed the stylet, and they found that as they did this, the radioactively labeled carbon-14 was found to be localized in this part of the plant only. It never went up this way, meaning they determined or concluded that plants are going to move phloem in a one-way direction from source to sink. This particular leaf would photosynthesize and move its phloem sap obviously in this direction. This leaf that they radioactively labeled obviously moved the phloem in this direction. These leaves would move its phloem in this direction, but it was a one-way flow. Water is going to be moving up the plant in a one-way flow. Obviously water can be recycled into the phloem and then pulled back into the xylem, but these radioactively labeled carbon compounds were really important in identifying how plants make phloem sap products, or how they make sugars, and then ultimately how they move those sugars through the plant. This is a really important experiment. You have to know it for the IB exams. Bring your questions to class. See ya.